is Dr. Michael Jackson, who is the Senior Vice President of the Conrad Prebys Center for Chemical Genomics, as well as the Director for the Center of Therapeutics Discovery. I will turn it over to Dr. Jackson.
All right, thank you. Can everybody hear me and see me? That's a yes. Okay, excellent. All right, so you're going to hear a number of words that all are actually the same thing. We call this I'm in charge of the Conrad Prebys Center for Chemical Genomics, which is really an academic drug discovery organization. We also call it the Prebys Center. And then to make it even more complicated, we've now transitioning to being calling it the Center for Therapeutics Discovery. But actually, they're all the same thing, right? So it's essentially about conducting work that's much more closely aligned to pharmaceutical companies and biotechnology companies within an academic environment. So is somebody controlling the slides? Yes, can I have the next slide? Keep going, we've already been through that, so let's keep, next slide. So the, oh no, go back one. That was too fast. So the objective of the Therapeutics Discovery Center really is transitioning from those understandings of what's gone wrong in a disease to how could I make a drug to impact that biology and impact a patient. And so we are very fortunate. We have invested, the Institute has invested over the last 20 years, millions of dollars in building a highly automated, state-of-the-art drug discovery center. It's mainly focused on small molecule drug discovery, i.e. pills rather than injectables. But that really doesn't limit most people in terms of their thinking of what to do. There's about 50 to 60 people in the center, all of whom, well, a great deal of whom have spent some time in biotechnology companies or pharmaceutical companies. Myself, I spent 15 years at Johnson & Johnson, at the Janssen pharmaceutical arm of that, doing drug discovery. The head of chemistry here, he's spent his time at Merck and Amgen. So there's a lot of expertise here that allows you to really understand how people from biotechnology and pharmaceutical companies approach a problem. And so it's not just the thinking of how to do that. Have next slide, please. It's the processes. And so the drug discovery process for small molecule drug discovery is a somewhat linear process that starts off with the thoughts of what could be the target of the drug, and then moves on through a series of steps in which you look to try and find a chemical that can modulate the activity of that target through typically either a very large chemical library screen, or more commonly at the or increasingly through more computational means. And we could, maybe I'll touch on that a little bit in a moment. So the strengths of the Prebys Center, which have been built up over the years, is really having these very large chemical collections and highly sophisticated robotics, and that can allow us to find a drug. And these are typically first-in-class projects. So a typical project that would involve postdoctoral scientists here is that you're in a lab, you come along, you are working on understanding the basics of a disease, and then you say, oh, look, what we need to do is develop an inhibitor to this particular protein. And that's a great place to come to the center, work with us in a collaborative way to try and figure out how do we do that. And in the process of that, you would get to learn many of the steps and stages and the process. Now, why is that relevant to you? Well, because way more than 50% of you who would come and do a postdoc here at Sanford Burnham Prebys will move on to industry. And what they are looking for is people with these skill sets and they're understanding not just of the hands-on experience of doing that, but the mindset. How do you actually go about it? How do you work in a team? How do you get to really understand this cross-disciplinary activity that is drug discovery? So over the next slide, the center has state-of-the-art robotics. So what does that mean? It means that all of the machinery that is doing liquid handling is done in a touchless, tipless way. And once you actually start using this technology, most experiments are being done in a 384 well format, or even more commonly now, a 1,536 well format. So you're doing 1,536 experiments every single with one shot in a plate. That becomes addictive, actually, because it gives you incredible power to give minimal error bars and to be able to really have very high quality data. This is a picture actually of the center. You can see in the foreground an echo access machine, highly capable robot, able of it just by that one robot you're seeing in the front called the Access, that's able to screen 150,000 compounds a day. We have 3 of those machines. In the background is the highest state-of-the-art. It's a imaging machine that can image plates, 1536 well plates, allowing you to do phenotypic screens of cells for almost any sort of phenotype you can imagine. Recently, the center, the next slide, please, has gone through a significant upgrade. And you can't see it all, but all the different pieces of the process have been upgraded. We've got now, as I say, 3 echo access machines giving you the power to screen hundreds of thousands of compounds in a day if you wish, or more typically do 3 or 4 different types of experiments going on in parallel. So giving a great deal of flexibility. And also allows you to be trained on one technology against cell-based screens, biochemical screens, and biophysical screens. So what's changing is, obviously, we talked a little bit that data science, but the next slide really talks more about computational modeling. Sorry, this was not computational modeling. This just shows you kind of what a 1536, 1534, well, that's something went wrong there. 1536 well plate, the super high density plates with little wells, allowing you to get very high precision data. And that's what allows you to really move forward a drug discovery project. Then the next slide, just wanted to talk about chemistry. We have about 15 chemists in the center with state-of-the-art capabilities. Again, really critical that you have people you work side by side with if you're more of a biologist. You want to befriend those chemists because without them, you will never come up with a drug. And you have to start learning their language and work very closely with them to be able to do that drug discovery process. And I say what's really changing, next slide, is in the last few years, the computational firepower to be able to do in silico docking and the ability to be able to find chemicals that bind a molecular target through use of structural biology and computational chemistry has really gone up a notch. You know, about 5 years ago, we had AlphaFold, and now we're starting moving into this way where compounds that bind a target, of course, dock and actually fit. And so we have now assembled, next slide, a whole series of tools that are computationally oriented, state-of-the-art tools here to allow you to conduct in silico screens and then calculate properties. In addition to that, the access to now on-demand chemical synthesis of billions of compounds, allowing with this more computational approach to be able to provide you an idea of what to make next and all of this is really accelerating at a pace that really is quite remarkable at the moment, and really is a place that I think many postdocs should spend some time getting exposed to. This is the future of the pharmaceutical and biotechnology world, and we are right there keeping up with it and trying to train people in these tools and the process. So I think that was kind of what I wanted to get through. In fact, there was a question about cryo-EM. So obviously one of the things that really allows you to do much of this molecular modeling and computational work is having a structure of your protein that is your target and also ideally your inhibitor or modulator bound to it. And with that, you're then highly enabled to be able to optimize that structure and improve its drug-like properties, not just potency and selectivity, but of course, you know, how it's going to be absorbed, whether it's going to be bioavailable, et cetera. So all those things are, this slide is actually from a postdoc, Merve, from the Prebys Center, who worked in collaboration with the scientific director of the cryo-EM core and has just published this work on the structure here. Okay, that was hopefully a bit fast, but hopefully got you through a little bit of what goes on in the Prebys Center.